Welcome to the story of a scientist who looked at a cob of corn and saw a revolution. Barbara McClintock didn't just study botany, she decoded the dynamic soul of life. While her contemporaries viewed the genome as a rigid, static blueprint, McClintock unveiled genetic plasticity, the radical discovery that genes could move, or jump, within the DNA architecture. Those variegated, speckled kernels aren't mere accidents. They are the visible evidence of transposable elements at work. This presentation chronicles McClintock's journey from a misunderstood pioneer to a Nobel-winning icon, exploring how her paradigm-shifting research forever changed our understanding of biological fluidity. It's a deep dive into the brilliance of a woman who truly heard what the plants were saying. Here, we witness the unwavering conviction that defines a scientific legend. Barbara McClintock stood alone against the rigid dogma of that static genome, armed only with her microscope and an inner certainty that others lacked. As a pioneering cytogeneticist, she didn't just map chromosomes, she discovered they were alive with movement. Her jumping genes, transposable elements, revealed a genome that was dynamic and responsive, not fixed in stone. Despite decades of skepticism from a scientific community that simply wasn't ready for her truth, she remained on the right track. This is the story of a 30-year wait for vindication, finally culminating in the 1983 Nobel Prize. It's a testament to the power of staying the course, no matter what the world says. To understand the scientist, we must first meet the woman whose independence was forged against the grain. Barbara McClintock's path was never easy. Her own mother initially vetoed her college plans, fearing that higher education would make her unmarriable. With her father's crucial support, she reached Cornell in 1919, only to find women barred from the genetics major. Undeterred, she used a genetics loophole, earning degrees in botany while mastering cytogenetics in secret. This slide highlights her defining trait, the capacity to be alone. This fierce childhood independence wasn't a burden, but a vital survival mechanism. It gave her the mental fortitude to endure decades of professional isolation while she mapped the hidden mechanics of life. McClintock's career was a restless odyssey for the one thing she prized above all: autonomy. At Cornell, despite mapping the physical exchange of genes, she was trapped in temporary fellowships with no permanent path for women. At the University of Missouri, the walls of bureaucracy and gender-based isolation further stifled her, yet she still managed to identify the critical breakage-fusion bridge cycle. Finally, she found her sanctuary at Cold Spring Harbor Laboratory. Liberated from academic politics and teaching burdens, she secured the total freedom she craved. For over 50 years, she was finally at liberty to simply ask the maize plant to solve life's mysteries, proving that true genius requires the space to breathe and the independence to listen. In 1931, Barbara McClintock and Harriet Creighton turned a theoretical mystery into a physical reality. For years, the idea of genetic recombination was merely a hypothesis. McClintock provided the proof. By meticulously observing chromosomes, she demonstrated the crossing over mechanism, the moment when chromosomes physically touch and swap segments like interlocking puzzle pieces during reproduction. This wasn't just a discovery, it was a bridge. By confirming Thomas Hunt Morgan's theories, McClintock effectively merged cytology and genetics, proving that the physical structure of a cell dictates the inheritance of life. She showed the world that genes aren't just abstract concepts, they are tangible, moving parts of a biological machine. Witness the destructive loop of the breakage-fusion bridge cycle. It begins with a snap, chromosomal breakage, leaving sticky ends that undergo improper fusion. During cell division, these fused strands stretch into a high-tension bridge until they inevitably snap again, birthing a new cycle of genomic instability. But McClintock's brilliance lay in identifying the missing piece. By studying this wreckage, she deduced that healthy chromosomes must require protective end caps to maintain stability and prevent such fusions. With incredible foresight, she predicted the existence of telomeres decades before molecular science could confirm her intuition. She didn't just map the damage, she envisioned the very architecture of cellular survival. Look closely at these kernels. They hold a secret that defied the laws of classical genetics. This was McClintock's great mystery. She observed color patterns, colorless, bronze, and mosaic, that shifted far too frequently to be explained by standard, random mutations. The paradox was clear. If every cell in a maize kernel contains the exact same genetic code, how could identical cells express such different colors? This slide explores the pivotal question that sparked a revolution. What was turning these pigment genes on and off during development? By questioning the anomaly in the maize, McClintock realized the genome was not a static blueprint, but a dynamic system, setting the stage for her groundbreaking discovery of transposable elements. Here we see the smoking gun of McClintock's revolution, the discovery of transposable elements. Think of a gene as an electrical circuit for pigment. In an intact circuit, the flow is steady, resulting in a solid purple kernel. But then, the impossible happens. A mobile element jumps into the sequence, creating an interrupted circuit that kills the color, leaving the kernel ghostly and white. The true magic lies in the restored circuit. As the kernel grows, that mobile element can jump back out. This late-stage departure allows descendant cells to regain their color, manifesting as the beautiful, mosaic spots that once baffled the scientific world. It's not a mutation in the traditional sense, it's a restless, living choreography within the DNA itself. Pulling back the curtain on the ACDS system, we find the regulatory engine behind McClintock's jumping genes. This wasn't just random movement, it was a sophisticated hierarchy of control. The activator, AC, serves as the master switch, an autonomous element that produces the enzymes necessary for transposition. Its partner, dissociation, DS, is the breaker that physically disrupts genes, yet it remains non-autonomous, powerless to move without AC's command. This was McClintock's core insight. Genes are more than a passive list of traits. They are part of a governed society where specific elements act as active controllers for others. By revealing this internal management, she proved the genome is a complex, self-regulating machine, not a static blueprint. In this final act, we witness the collision of two fundamentally different worlds. On the left lies the 1950s scientific dogma, a rigid, static landscape where genes are immutable beads on a string, fixed in place and subject only to rare, random accidents. But look to the right, and you see the world through McClintock's eyes. This is the dynamic genome, a fluid, vibrant mosaic where genes are not prisoners of their location. Through transposition, these mobile elements actively navigate the chromosome. McClintock's radical insight was that the genome is an active, responsive participant in life, intentionally restructuring itself to survive environmental stress. It is a transformation from a passive blueprint to a living, breathing engine of adaptation. Imagine standing before your peers, unveiling the secret to life's fluidity, only to be met with stony silence and whispers of madness. This was the summer of 1951 for Barbara McClintock. Her revelation of jumping genes was so far ahead of its time that the scientific community simply could not, or would not, understand it. Bruised by this hostility, McClintock entered the blackout in 1953, choosing to stop publishing her transposon data in scholarly journals. Thus began the silent decades. Yet, silence did not mean surrender. For nearly 20 years, she remained at her microscope, trusting her eyes over the consensus. Guided by an unshakable feeling for the organism, she continued her work in private, waiting for the world to catch up. Even after her revolutionary discovery of transposons, Barbara McClintock continued to push the boundaries of biological thought. Here, we see her predicting the field of epigenetics through her work with the suppressor-mutator system, proving that genes could be silenced or activated without altering their underlying code. Her vision also expanded globally into the field of ethnobotany. For nearly a decade, she traversed South America, studying how the chromosomal evolution of maize was inextricably linked to indigenous agricultural history. Whether at the microscope or in the fields of the Andes, McClintock remained decades ahead of the curve, seeing patterns of inheritance and evolution that the rest of the world was only beginning to imagine. The long wait for vindication finally ends as the rest of the biological world catches up to McClintock's radical vision. What began as a solitary, misunderstood observation in maize during the 1940s slowly rippled through the entire kingdom of life. By the 1960s, bacteria revealed genetic regulation and transposons carrying antibiotic resistance. In the 70s, molecular biologists finally isolated and cloned these elements in yeast and fruit flies, providing the undeniable chemical proof McClintock had lacked for decades. Today, the truth is staggering. Nearly 50% of the human genome consists of these jumping genes. This slide is the ultimate validation, a final confirmation that Barbara McClintock wasn't just right about plants, she was right about the very essence of humanity. The curtain rises on the final act of a long journey towards scientific justice. After decades of being sidelined by a skeptical establishment, Barbara McClintock's brilliance finally received the global recognition it deserved. This slide chronicles her ultimate vindication, beginning in 1970 as the first woman to receive the National Medal of Science, followed by the MacArthur Genius Grant in 1981. But the crowning moment arrived in 1983. At age 81, McClintock was awarded the Nobel Prize in Physiology or Medicine. To this day, she remains the only woman in history to win an unshared Nobel in this category. It was more than a prize, it was a standing ovation for a pioneer who trusted her eyes when the world chose to look away. We conclude our journey with the enduring legacy of a woman who saw the world not as it was dictated to her, but as it truly functioned. Barbara McClintock stands today as the founding mother of modern genetics, a pioneer who proved that a feeling for the organism is as vital as the microscope itself. Her story is the ultimate masterclass in scientific integrity. She didn't chase consensus, she chased the truth hidden within the maze. By bridging rigorous logic with deep intuition, she transformed our understanding of life's fluidity. Her life remains a powerful reminder that the most revolutionary discoveries belong to those brave enough to stand alone until the rest of the world finally catches up.